Hello children, this is a biology class for class 10, all sections 10A, 10B and 10C. We are in chapter number 3, that is genetics. Since the chapter is too long, we cannot complete it all at a time. So here, today, I'll be discussing on the subtopic of the chapter number 3, that is Mendel's experiment on inheritance. Mendel's experiment on inheritance. Okay? Gregor John Mendel, who is an Austrian monk, okay, was the first person who founded the basic principles of genetics. Okay? He was the first person to discuss on the genetics, alright? Based on the idea of genetics given by the Gregor John Mendel only, we have now a lot of ideas about genetics now at this present time, okay? We have a lot of ideas about genetics now, we have a lot of applications of genetics, but it is all rooted to the idea given by Gregor John Mendel. That is the reason why Gregor John Mendel is also known as the father of genetics. Okay? Father of genetics. So Gregor John Mendel is famous for his two crosses or for his two experiments. So they are monohybrid cross and dihybrid cross. Monohybrid cross and dihybrid cross. He did his experiment. He did his this cross, okay, on a plant called pea plant. Okay, pea plant which was being grown in his own field, in his own garden. So he did his experiment on the pea plant. Okay, pea plant, on the pea plant. Okay, whose botanical name is Pisum sativum. Pisum sativum. Alright? Pisum sativum. He did his experiment on this plant called Pisum sativum or pea plant. Alright? So here, as I already told you, that he did it in 2 ways. I mean, he is famous for 2 experiments, that is monohybrid cross and dihybrid cross. Alright? So monohybrid cross is the one where he considered only one character at a time in his experiment. Since one character only is taken in consideration, the cross is called the monohybrid cross. And whereas the dihybrid cross is the one where the 2 characters are taken into consideration at a time in the experiment. That is called dihybrid cross. Okay? So today we will be talking about the monohybrid cross only. Alright? Monohybrid cross. Then if the time permits, we can even do the dihybrid cross today itself. Otherwise, we will be mainly talking about the monohybrid cross today. So for monohybrid cross, he considered the different characters of the pea plant. Alright? What are the characters he considered for his monohybrid cross? You can see all out here. See. Character, and then you can see dominant recessive trait. Character, flower color. He considered, flower color that is purple and white for his monohybrid cross. And another experiment again in the monohybrid cross itself, he considered seed color also, where yellow and green color of the seed was there. Then again, seed shape, where the shape of the seed, that is round shape of the pea, and the wrinkled shape of pea was also considered in his monohybrid cross. And pot color, green and yellow, pot shape infinitely constricted, flower portion axillary and terminal. Axillary means the flower which is in the axil, whereas terminal means the flower on the top of the plant. So that also he considered in his monohybrid cross. And plant height, that is tall and broad. This height of the plant, pea plant also, he considered in his monohybrid cross. Alright, 7 different characters he considered for his monohybrid cross, and whatever the result he got in all the experiments, elements were the same. Alright, were the same. Alright, so here let me tell you character, color. Let us say color. So in the color or for the particular character, there is a gene which we have discussed last time also in the last time's online class also we have to talk about it. For that character, there is a gene having two different alleles. One allele is for purple color here, the other allele is for white color here. One is dominant, the other one is not, the recessive. Alright? One is dominant, the other one is recessive. In these two colors, purple is always dominant. Okay? The other one is that is the white, that is recessive, dominant and recessive. So this is the character and these two are the traits. For each trait, one separate allele is there, which is together. One allele for this, the other allele for this, which are together in that particular gene, which was discussed in the last examination also. Similarly, in the case of seed color also, yellow and green, yellow is dominant over the green. For that also one is there, one gene is there having two different alleles. One is for yellow, the other one is green. So in this case, yellow is dominant on the green. Alright? In the same way, seed cell also. Seed cell. For seed cell is a character for which one particular gene is there having two alternative forms which are called traits. So for which where one allele is for round, the other allele is for greater. In the same way, you find out to this plant height. So last time you see the height, plant height. One particular gene for the height of this plant, which have got two traits, that is tall and short, for which two alleles. One allele is for tallness of the plant, the other allele is for the dwarfness of the plant, where tallness is dominant. So these seven characters he considered for his monohybrid cross. As I already told you that in all the experiments he found the same result. Okay, so out of this seven, let us take one character so that you understand how he did it, how he went about the process, how he went about the experiment. For which, let us consider one of the characters. Let us consider this last one, that is plant height for his monohybrid cross. Today we will talk about monohybrid cross. Monohybrid cross. For monohybrid cross now, let us consider the plant height today. Plant height. Alright? Plant height. So two plants. That is one pure tall, the other one is pure dwarf. Alright? Tall plant and dwarf plant. Tall plant and dwarf plant. Alright? So tall plant is represented by capital T and this dwarf plant is represented by small t. So now you maybe question yourself, why this double T, capital T are there, why the double small t are there? As I already told you, that tall plant has one particular gene having two alleles. So both the alleles in this case are same, that is homozygous dominant. Both are dominant, homozygous dominant. That's why it is pure tall. Whereas on the other side, there is a gene having two alleles which are recessive. Both are recessive, small t and small t. Alright? So this is how it is done. Okay, this is how it can be represented. Okay, how he went about his cross, how he went about his experiment. What it is that, what Mendel did in this case is that he crossed the dwarf plant with the tall plant. So how he crossed it? You might be questioning yourself again. So how he crossed it? Okay. So for that, we do this thing. So this is, alright? Okay. This is N terminal. and this is the stigma. Alright? So, one thing you please remember that this pea plant, this Pisum sativum, is a complete flower, is a bisexual flower where self-pollination is possible. Alright? Self-pollination is possible. But here, Mendel was going to cross these two varieties of the pea plants. Okay? He was crossing these two varieties of the plants. So where cross-pollination only was, he just wanted to make the cross-pollination in these two varieties of the plants. So for which, what it is that he collected all the anthers from here. Anther from this plant. Anthers means the place where the pollen grains are produced. He collected all the anthers from here. Anthers, okay? From here, which bears pollen grains. So he collected all the anther and transferred into this stigma. Which is called cross-pollination. Okay? Cross-pollination. He transferred here. Alright? So, and now, in order to check the self-pollination, since androecium is there, or anther is there was a chance of the transfer of the pollen grains from here to here also, which is self-pollination. So, in order to check the self-pollination out here, he removed all these androeciums from here. He removed. Whichever the flowers were there in that plant, he from there, all the androeciums he removed so that there was no self-pollination. Alright? So, he wanted to have the cross-pollination. He wanted to mix these two varieties of this pea plant so that he wanted to see the inheritance of characters from parents to the offsprings in the next first and second generations. Alright? So he crossed like this. Okay? When he crossed like this, so that there was no self-pollination. Alright? Then what happened? He allowed the plant to grow. This plant grew and produced lot of seeds. And he collected those seeds and sowed. He sowed. Then from those resulting seeds, what kinds of plants were produced? Okay? What kinds of plants were produced? He saw all the plants having been produced were all tall plants. They were all tall plants. All the plants produced in this generation were all tall. Okay? So these were the tall plants. What kind of tall plants? Hybrid tall plants. All the plants produced in this generation were tall plants. They were hybrid tall plants. And he named this generation as first filial generation, F1 generation. Or first filial generation. Alright? So that means this resulting series produced all the plants tall. Alright? They were tall. But what kind of tall? Hybrid tall. Alright? Then he allowed the plants to grow. Alright? Then he collected the seeds again and he grew it again. And allowed them to self-pollinate. No cross-pollination now. Allowed them to have a self-pollination among their seeds. Okay? Then what he observed in the second generation, let us see now. Right? Okay, when he allowed the plants of the first generation for the self-pollination, he found the plants like this. He found the plants like this. He found the plants like this. That is, out of the four plants, the resultant plants were like this. Those plants which we found in this generation were named as the plants of second generation or F2 generation. That is second generation. Okay, this is first generation and the second generation. So in the second generation, when he allowed the plants for the self-pollination, he found the plants like this. So they were all tall plants, and this only one was the dwarf plant. Okay, out of 4 plants, 3 were tall and 1 was dwarf. Alright? So if you see the phenotype, of this phenotypic ratio means ratio of the plants which are seen, how it is manifested. So 3 kinds of plants, these 3 plants were all tall. 3:1. 3:1. That means 3 tall plants and 1 dwarf plant. So the ratio was 3:1. That means 3 tall and 1 dwarf. But genotypic ratio, if you see the genotypic ratio, you will find the ratio like this. 1:2:1. See, 1:2:1. That means 1 is pure tall. Alright? Then 2 are hybrid tall. And last one is pure dwarf. So this was the result in the F2 generation. Okay, in the F2 generation. So this dwarfness was not seen in the first generation, but the plants which are in the first generation also were carrying the allele or the dwarfness of the plant, which was seen in the second generation. From every 4 plants, 3 were tall, 1 was dwarf. Alright? So out of these 3 also, 1 was pure tall and next 2 are hybrid tall. And the last one is the pure dwarf plant. So that was the result of this one. If I have to explain this phenotypic ratio again, we find like this 3:1, this means 3 tall plants and 1 dwarf plant. So genotypic ratio is 1:2:1. That is 1 pure tall, 2 hybrid tall and 1 pure tall. Okay? So that is genotypic ratio 1:2:1. Alright? So now same can be shown by the Punnett square method also. By Punnett square method, like what we have discussed last time also. Same can be shown by the Punnett square method. Alright? Let us see now. This tall plants and it is dwarf plant. Alright? So okay. So if you cross this one here, this comes here, this comes here, this comes here, this comes here. Alright? So if you cross it, T and TNT, right? Then this TNT, this TNT, then TNT. So in the F1 generation, all are tall. Okay, all are tall. Hybrid tall. hybrid tall, like this. In the F1 generation, all the plants are hybrid tall. Okay. Then when it is self-pollinated, when it is self-pollinated, let's take on here from here. When it is self-pollinated, so one tree comes out here, another tree here, this tree here, this tree here. If you crisscross it, you find the result like this. Okay, this is F2 generation. F2 generation. Okay? If you see the phenotype, here you see 1, 2, 3, 1, 2, 3, all three. 3 is to 1. That means 3 tall, and 1 dwarf, and 1 dwarf. That is phenotype. So out of 4 plants, 3 are tall plants and 1 is dwarf, like this here. Alright? Then genotype would be 1 pure tall and this 2, this and this, hybrid tall, and last one is dwarf. That is genotype. That means 1 pure tall, 2 hybrid tall, and last one is 1 pure dwarf. Okay, so the result is the same. Okay, result is the same. Whether by this method or by this Punnett square method, result is the same. Out of 4 children, out of 4 plants, so 3 are tall, 1 is dwarf. Alright, the same result is the same in both the methods. Alright, so this is what we have found in the monohybrid cross done by Gregor John Mendel. And I think this is enough for today, because it is you need to give your time, you need to give your attention to understand it more. That's why it's better to end it up to this only. So the dihybrid cross, which I was telling you before, the dihybrid cross we'll do it in the next session. Alright? So since I have finished this first monohybrid cross out here, giving right from A to Z. Alright, I have tried my best to make you understand about monohybrid cross. You please try to practice this, inheritance of these plants when the cross was there between the 2 varieties of this pea plant, taking the character of tall plant and the dwarf plant. Whatever we have found, please try to do this at home. You practice it as a home assignment that I am giving you. Not only that, you do the other one also. There in the book, you can see page number 31, where the same monohybrid cross you see being done by, having done by Gregor John Mendel, taking the character of the 2 varieties of the plant. One having having the terminal flower, the other one having the axillary flower. Whichever we have done here, you please do this also at home. So these are the 2 homeworks that you have to do at home. So try to understand, please try to go through the book given, try to go through the works done in the book also, and if it is not clear to you, you please let me know. If any doubt is there, you can contact me on this number also. This is my number, 9793331991. This is my number. So I have got WhatsApp also there on this number. So you can contact me on this number. You can WhatsApp me also whatever the problem you have got. And still, you are not clear with the work that I have tried to make you understand, then we can do it afterwards whenever we meet. I am still optimistic that we'll meet again very soon. So when we meet, then we'll do this once again. Okay? Thank you so much.